So we left the lecture 2 days ago with the photosynthesis irradiance relationship. When we plot the rate of photosynthetic oxygen production or consumption, which is net photosynthesis on the y-axis, and against light, we get these relationships with respiration happening in the dark, a compensation point where production and respiration balance each other at or zero, some maximum photosynthesis, initial increased slope, and photoinhibition if light gets really high. And then we have high light and low light adapted species. And that brings us to the first question of the day. If you're adapting to low light as a photosynthetic organism, and it could either be long-term evolutionary adaptation or short-term changes in physiology that lead to this, but there could be some evolutionary trade-offs in this that make it so you can't be both high light and low light. So can you list some potential energetic costs adapting to low light and some for adapting to high light? So some of the things, energetic costs that might be to low light is that you have to produce more chlorophyll, as one of the students said in the chat. You basically have to cover more of your cell surface with photosynthetic units and things to collect light and to photosynthesize, which means that your cell membrane can't be covered by things like as many proteins to take up nutrients. So you're basically going to have other functions that you have to put energy towards doing that, and that'll take that away. That'll take that away. In contrast, if you're in high light, you've got all these energetic electrons, and you can actually be photoinhibited. And so you need to cut back on your chlorophyll, but the high light also can cause problems. And we'll talk about this in other portions of the class, like extreme environments. But you may even need to have extra pigments that protect you from those energized electrons and capture light before it does damage to you, since you don't need it all if there's high light. So that can cost you energy. For example, these are carotenoids that algae produce to protect themselves, and then zooplankton will eat them. And some of you maybe have seen trout from high mountain lakes that are pink skin, pink flesh, or salmon from open ocean that have pink flesh, and that's because they eat the zooplankton that have a little light protective carotenoids that they've gotten from the algae, and it just magnifies in the food webs. And we'll see things like watermelon snow and things like that that are related to ways that organisms can deal with high light, because you have to do something to deal with that extra potential energy and damage. Other factors can influence photosynthetic rate as well as light, including temperature. And so if we have net primary production, and this is carbon, based on carbon as a function of temperature, we can see that there's a whole bunch of studies that have been synthesized here, but there's an upper envelope. And that's what the maximum potential photosynthesis is, as limited by temperature. And we'll talk about temperatures in several times in this class, but essentially you have an exponential increase in the rate related to temperature, rate of metabolism related to temperature, which is known as the irradiance plot. And you can get at what are called Q10s, the amount of increase, the number of degrees of you have to get a doubling in the rate. And roughly, rule of thumb is they tend to be around every 10 degrees C or so, you get a doubling in rate. This curve appears to be slightly higher than that. When we go from 10 to 20, we're going from about, well, maybe 75 to 150. It's not too far off. So that's about it. What are some reasons for these? So you're not always at that maximum potential. Why are there these measurements from different environments that the photosynthetic rate's relatively low, even though the temperature's high? Can you think of some reasons that might be related to that? Anybody? Well, we can think of something like Tuttle Creek Reservoir. Water availability, well, maybe, but we're in freshwater. So these are all freshwater organisms. So nobody's going to be limited by water unless they're maybe in a hypersaline situation where water can be limiting in a sense. So not water availability, but it could be something else. The amount of nutrients, right? We could have an ultra oligotrophic system like Lake Tahoe or Crater Lake, where the nutrients are super low. So it doesn't matter how much light, how much temperature there is in the water. It's just going to be that. So nutrients, what was the factor we talked about just before this that could influence photosynthetic rate? Anybody?
Suspended solids.
Yeah, light. So suspended solid. So we've got, for example, Tuttle Creek Reservoir that has real high turbidity nearby here. And the chlorophyll levels and the photosynthetic rates are very low, even though the nutrients are quite high. So there's a variety of things that can shut down photosynthetic rate. But then there's these upper limits. And you can think of a series of upper limits related to that. There has to be enough light. There has to be enough nutrients. The temperature has to be right. Maybe there would be grazing or toxins that knock the biomass down that could drop the photosynthetic rate. And we have another factor here, water velocity. So this is a series of measurements of phytophthora, which is a green alga, filamentous green alga that attaches to the surface. And the photosynthetic rate of that organism as a function of water velocity. And this is again a carbon fixation rate measured by isotopes and radioactive isotopes. And we see that the average down here is around 20 at 0 water velocity. But as the water velocity increases to 25 centimeters per second, so this is a pretty brisk flow of stream right here, the photosynthetic rate goes up and up. So why might that be related to the diffusion boundary layer? Okay, so the first question is what happens to the thickness of the diffusion boundary layer as the water velocity increases? Does it get thicker or thinner? Boundary layer gets thicker. Well, you had a 50% chance, Lane, of getting that right, and you didn't. So, no, it gets thinner. The faster the water flows, if it's flowing across a rock, right, the faster it flows, the closer that flow is going to get to the rock or whatever's on the bottom. And so you'll have turbulent flow moving further and further down, right? The no slip zone will get thinner and thinner. So what's going to happen to the diffusion rate? Is it going to increase or decrease? Because remember, the no slip zone is where molecular diffusion predominates. Molecular diffusion is slower than the turbulent transport. Right, it increases. The diffusion rate increases. So why would a rate of increase of diffusion increase the photosynthetic rate? And so there's reactants and there's products from photosynthesis. They can absorb more CO2, right? So maybe CO2 is limiting. There's CO2 dissolved in the water, and we'll talk about that. So the rate of carbon dioxide flux into the photosynthetic organisms that are on the bottom could be greater with more diffusion and higher water velocity. What's one of the products of photosynthesis? One of them is organic carbon, but what's the other product of photosynthesis? Oxygen, right? Well, oxygen is a toxic molecule, it turns out. It reacts. It likes electrons. It'll react with a lot of things. It's actually a waste product that needs to be dumped off, and super high oxygen levels can cause problems with organisms. And essentially, even us, even though we need oxygen for respiration, we control oxygen very closely in our cells. And if oxygen really gets into them, other than coming in via hemoglobin, it'll kill the cell. But if it comes in the hemoglobin, that gets transferred where it needs without being just free in the cell to react with everything, we're okay. So you can also dump off oxygen. And then the other thing what's another thing that might limit photosynthetic rates? Temperature is not it, but we had one more factor that we talked about that might limit photosynthetic rates in a prior discussion. So to build cells, organisms don't just need carbon dioxide. What else do they need? Nitrogen, good. Phosphorus, right? All the elements that we'll talk about that are needed. And so diffusion inward of the nutrients required for photosynthetic growth. And I'm muting you, Logan. Okay. So one of the things that's happened over time is that because of chlorofluorocarbon increases, historically up through the 80s and 90s, the chlorofluorocarbons went into the upper atmosphere and destroyed ozone, O3. Ozone is important because it shields ultraviolet from reaching the Earth's surface. So that's why when cyanobacteria first had the evolutionary adaptation of photosynthesizing, splitting water, creating oxygen, they oxidized the Earth's atmosphere, led to oxygen, and shielded essentially all the habitats on Earth from the UV. So shallow surface waters and terrestrial habitats became much more habitable once that shield was there. When we started using chlorofluorocarbons as refrigerants and also as propellants for aerosols like spray on deodorant and stuff like that, they're very non-reactive molecules, but they'll float up into the upper atmosphere, but they will convert ozone back to oxygen, so they'll deplete the ozone. They created large holes in the ozone around the poles, and particularly around the Antarctic. And so there was a very rapid Montreal Protocol. There was rapid agreement on limiting their use, and they really did cut back seriously globally on their use. That's one of the big environmental one of the big environmental success stories in terms of global environmental problems. But because of that, it spurred a lot of research on what the increased UV would do to the natural systems. Some of the things that happened were pretty predictable. Other things that weren't. Other things were less predictable and less direct. I should mention that the ozone hole now, so that happened in the 80s when they started clamping down on production of chlorofluorocarbons. The ozone hole in the Arctic is just starting to close up now, statistically. So with global environmental issues, it takes a long time for the system to respond to changes. And then to add on to the problem, nitrogen pollution is causing nitrous oxide. We'll get at the mechanisms of that to enter the atmosphere at greater rates, and that's decreasing the ozone throughout the entire world, in addition to it being a greenhouse gas. So decreased photosynthetic rates of phytoplankton and periphyton in high altitude lakes. So they're getting exposed to more UV already, and if you up that even more, they're going to get more effect. Lower nitrogen uptake rates, lower populations of consumers, of the producers. So this is an interesting experiment here. Max Bothwell had these experimental stream channels and put UV filters to knock the UV out on some of them. And over the short term, the algae really took off with lower ultraviolet light. But over the attached algae, the periphyton, but over the long term, all of a sudden the algae went way down, and they couldn't figure out why. And the reason is the grazing invertebrates that ate the algae were able to reproduce more successfully in low ultraviolet light, because the ultraviolet was harming the eggs of those organisms. And so when the eggs had less UV effect, then there was higher population to build up, and ultimately production went down. So that's an indirect effect of ultraviolet on the food web, not a direct. And so there's a whole bunch of other things, more ability of cyanobacteria to fix nitrogen. If there's a lot of dissolved organic carbon, it absorbs UV in the stream or in the lake. So there's not so much problem. It hurts mosses. Changes the community structure so you can see all these things that have effects. Some of them could be globally. So growth of sphagnum in a bog. Peat bogs are a big sink of organic carbon globally, and so this could have an effect in the long term. And interacts with other things. So if you had acid precipitation on lakes, if they're being hit by one stressor, and if you put another stressor on top of that, ultraviolet, it could have more than additive effects on it. Okay, And so that's photosynthesis. And then we've talked about photosynthesis and respiration, balancing out where oxygen is. And here's just some examples that we have of spatial variation in oxygen at two different scales. And the one on the left is wells that have been drilled down to 8 meters. And we used microelectrodes to measure what the oxygen concentration was in the soil core that we took out of each of those. And we see that under cropland, there's higher oxygen with depth than there was under prairie. And that's because there's a lot more organic carbon in soils and in natural grassland soils. And in cropland, because they're continuously tilling the soil, the respiration rates are higher. It makes the carbon more available, breaks down the aggregates, makes the carbon available to the microbes to break down. So the carbon's lower, the respiration rates are lower, so the oxygen's higher. And that varies some with depth, where the highest rates of microbial activity are near the surface, and so the oxygen's the lowest there because they're using that up. And this is a contrast. This is a leaf on the right-hand side, and it's just, these plots are just, it's just an XY plot. And what the student did here was take a leaf and put an oxygen microelectrode down in the leaf, went over a millimeter, went over another millimeter, or half millimeter this time, I think. And did that, and then just did a grid to measure the oxygen. And we see that the parts of the leaf that had the highest oxygen, they were at 60%, 70, 50% oxygen, were on the rib of the leaf, right? And that's recalcitrant stuff. So that does not get broken down very easily. The rates of respiration are low. The microbes can't be very active there. But if we get into the more fleshy part of the leaf, if you will, the part that doesn't have that structural material, but the actual active part of the leaf, of the dead leaf, that has more carbon available, is less recalcitrant, and the respiration rates are higher. So the oxygen is lower, and you can see that there's zero oxygen in the places even on the surface of the leaf. So if you go down in the leaf, you'll get anoxic zones. So there's sort of two spatial scales that we're talking about here. We can talk about temporal variation in oxygen and coupled with spatial. And there's a couple examples on here. The first one is just groundwater spring. And we see the oxygen's really constant there. It doesn't change very much, right? It's just really pretty rock solid over time. But if we look at an open lake or stream, we have higher oxygen during the day and lower oxygen at night. Why do you think that is? What do you think leads to higher oxygen in the day and lower at night? Photosynthesis, good. Photosynthesis, respiration's always going on. Photosynthesis happens during the day, but not at the night. Both photosynthesis and respiration during the day, but just respiration at night. So the oxygen goes up during the day, here, and it goes down during the night, here. And then the lake, remember the pelagic zone is the center part, right? And the littoral zone is near the edges. And this particular lake had a really dense macrophytes. So why do you think the oxygen swung so much more in the littoral zone than the pelagic zone? And hopefully everybody's putting their name in and answering some of these questions so we can make sure to get that on the question of the day list. So dense macrophytes as opposed to plankton. What do you think? How do you think that relates to the bigger swing of oxygen in the littoral and panel B than the pelagic? Right, Tom, photosynthesis does cause O2 spikes during the day in the littoral zone. That implies, though, that there's more photosynthesis, right? You get a bigger burst of photosynthesis. So that would suggest that the total photosynthetic rate is high when you have a lot of macrophytes. So if you have dense plants, you're going to get more photosynthesis. And then somebody threw wind in there, I believe, and that could be it as well. It could be that those dense macrophytes keep the mixing from the wind down a little bit, and you're close to the edge, there's less. So maybe there's equilibration of oxygen with the atmosphere in the open part that's faster than in the littoral zone. So there's a couple of things. There's a couple of things there. In addition, it could be that this is a somewhat turbid lake. So maybe there's a bunch of organic carbon coming in from outside that the plankton is dominated by heterotrophs and not autotrophs. The last one here is panel to talk about. Well, there's a couple more. So the diurnal trend in oxygen on the streams here. This is what we were using in this experiment that I was just talking about from Mongolia to estimate the photosynthetic rate. So if you can get what the aeration rate is, the exchange rate with the atmosphere, and some other physical characteristics of the river, you can actually model what photosynthesis and respiration are from data like this. And Odom, who's a famous ecosystem ecologist, came up with this idea for streams in the 50s, but it's one that's developed over time, and it's gotten a lot easier to do because we have oxygen, logging oxygen sensors that are fairly stable and accurate now. So we can collect a lot of data on that. And I've mentioned a little bit microelectrodes, and this graph down here is just a graph from a dense cyanobacterial map that was taken from the Saskatchewan River. And the light is here, and I just put shade over it, and you can watch at the time within a minute the oxygen dropped substantially. And then I pulled the shade off and the light went up. And you can actually use this to calculate what the photosynthetic rate is at a very fine spatial scale within these maps. So you can imagine these little millimeter thick mats have screamingly high oxygen when they're photosynthesizing, and then at night it just drops down to nothing at all. This was for a workshop that I was involved in on the river, and I was the person that brought the microelectrodes, and I couldn't get the darn things to work. And it was really frustrating because all these experts from around the world on different parts of photosynthesis were there, and we were going to do this experiment where we did all these different ways to measure it. And I couldn't get the electrodes to work, and I was swearing under my breath and messing with all the connections and everything like that, and finally just gave up, and we all sat down and started eating lunch. And I just left it sitting there, and I wasn't paying much attention. And I looked over at the graph, and a cloud had come over, and the oxygen just went down with lower light, and then the cloud went away and it popped back up. I'm like, hey guys, it's working. So this actually was brings back that story to me. It also makes the point that the oxygen can be very dynamic in some of these systems.